My name is Tracy Papenfus, and I am a member of the Health and Environmental Sciences Institute Immunosafety Technical Committee, or HESI ITC. The HESI ITC is a scientific organization whose members hold positions in industry, academia, and government, and who work to assess immune safety and its impact on human health. This presentation has been prepared for you to introduce you to the field of immunosafety and its role in the development of drugs, therapies, and vaccines. In November of 1961, thalidomide was taken off the market due to thousands of birth defects and deaths seen in children. Thalidomide was prescribed for the treatment of morning sickness, but it was its immunomodulatory and anti-angiogenic activities that contributed to the tragic results and highlighted the importance of stringent safety assessment procedures and guidelines. In 2006, a monoclonal antibody that was designed to manipulate the body's immune system to treat leukemia and autoimmune diseases was given to 6 healthy volunteers during initial phase 1 clinical trials with disastrous results. Life-threatening side effects, including cytokine storm and multi-organ failure, highlighted the importance of understanding immune differences across species and changed the approach when moving from preclinical safety studies to clinical trials. In 2020, a novel coronavirus and its associated disease, COVID-19, caused a global pandemic infecting over 60 million people and resulting in nearly 1.5 million deaths. In response, there was an unprecedented scientific effort to understand, treat, and develop a vaccine more rapidly than ever had occurred in history. In November of 2020, several promising vaccine candidates were developed that appeared to be effective, but people still had questions about whether safety had been sacrificed for speed. The question on many people's minds for COVID-19 vaccine development was, how can we get it done right and get it done safely and effectively in only months when this process normally takes years? Several factors contributed to the rapid development of COVID-19 vaccines without shortcutting safety. One is that the knowledge that the spike protein is important both in pathogenesis and in the development of immunity. We also have the technology and scientific capabilities that build on our knowledge of previous diseases, platforms, delivery methods, manufacturing, and known safety of these modalities. Scientific and manufacturing activities that have traditionally occurred sequentially are occurring in parallel or simultaneously. Time spent evaluating go, no-go decisions and financial considerations have largely been removed, resulting in overlapping phases and shortened development time. Finally, the urgency of this pandemic has resulted in collaborations and partnerships across the globe and impact the determination of benefit versus risk. The development of a vaccine, such as COVID-19 vaccine or any other drug or therapy, requires the development of something that works but is also safe. For therapies such as vaccine that directly affect the immune system, it's particularly important to understand that these effects to be able to evaluate benefit versus risks. This is immunosafety. Immunosafety is a component of safety evaluation required by regulatory authorities that focuses on how new drugs, therapies, vaccines, and chemical agents affect the immune system. Immunosafety evaluation occurs throughout drug development across many disciplines and not only determines overtly toxic effects, but identifies the spectrum of potential immune effects of any therapy. So for a new therapy under development, in addition to determining whether it works, is to determine whether it is safe. The evaluation of expected and unexpected effects help determine whether the benefits outweigh the risks. Such decisions take into account many factors, including, but not limited to, the disease, current therapies, toxicity, and patient population. Therapies that can affect the immune system can do so simply by the nature of the therapy or compound or can be specifically designed to modulate the immune system. Here are just some of the immunomodulatory therapies that are available, and these various therapies that have numerous applications in a variety of diseases, some of which are listed here. Over the past decade, there has been a relative explosion in the number of therapies that can influence the immune system, thus increasing the need and importance of immunosafety evaluation. Assessing immunosafety involves a collaboration among scientists and the integration of data across many disciplines. Immunotoxicology and immunopathology, as well as clinical monitoring, all occur throughout drug development process to provide information that facilitate a detailed understanding of the immune system changes in the context of therapy, relevant disease state, and the variability of an individual's immune response. As you can imagine, immunosafety work involves numerous professionals working together from bench to bedside. A whole host of scientists, researchers, medical professionals, and regulatory personnel are involved and work collaboratively. The careers and training are diverse, and it is the different expertise and perspectives that help provide an integrated understanding of immunosafety for any therapy. In summary, immunosafety is a dynamic field that evaluates novel biomedical research advances and addresses public health concerns to determine the effects on the immune response and weigh the benefits versus the risks of new therapies. Ultimately, determining immunosafety for any given therapy, disease entity, or in any patient is a coordinated and collaborative effort by scientists, medical professionals, and regulatory personnel to help provide safe drugs and therapies. Thank you for your time and attention.